Welcome to Cancer 101, the podcast where we dive into the intricate world of cancer biology. I'm your host, Ritika, a neuroscience postgraduate with a passion for understanding the cellular and molecular mechanisms underlying diseases. Today, we are exploring how genetic and epigenetic alterations contribute to cancer initiation and progression. Joining me is Aditi Dubey, a brilliant student and emergent expert in cancer biology. Aditi, welcome to the show.
Thank you, Ritika. I'm excited to be here. Let's dive into this topic. Cancer's evolutionary journey reads like a gripping detective novel, filled with unexpected twists, hidden clues, and cunning culprits orchestrating the chaos.
Let's start with the basics. Aditi, could you explain the role of oncogenes and tumor suppressor genes in cancer development?
Certainly, oncogenes are mutated forms of proto-oncogenes, which normally regulate cell growth and division. When these genes become overactive, they can drive uncontrolled cell proliferation. A classic example is the RAS gene, which, when mutated, leads to continuous cell division signals. On the other hand, tumor suppressor genes act as the cell's brakes. They regulate cell division, repair DNA errors, or initiate apoptosis. When these genes are inactivated, cells can grow uncontrollably. The TP53 gene, often called the guardian of the genome, is a well known tumor suppressor. Mutations in TP53 are found in about 50% of human cancers.
Moving beyond genetic mutations, epigenetic changes also play a significant role in cancer. Can you shed some light onto this?
Absolutely. Epigenetic modifications, such as DNA methylation and histone acetylation, regulate gene expression without altering the DNA sequence. In cancer, abnormal DNA methylation can silence tumor suppressor genes, while histone modifications can either activate or repress gene expression. These changes can disrupt normal cellular functions and contribute to tumorigenesis.
It's fascinating how cancer cells can hijack developmental pathways. Could you provide examples of such pathways involved in cancer?
Yes, two prominent examples are the WNT and Hedgehog signaling pathways. The WNT pathway, crucial for embryonic development, is often aberrantly activated in cancers like colorectal cancer. Mutations in components like APC lead to uncontrolled cell proliferation. Similarly, the Hedgehog pathway, essential for cell differentiation, is implicated in cancers such as medulloblastoma. Mutations in genes like PTCH1 and SMO can result in continuous pathway activation, promoting tumor growth.
Given the role of epigenetic changes in cancer, are there therapies targeting these modifications?
Indeed, epigenetic therapies aim to reverse abnormal gene expression patterns. Histone deacetylase inhibitors like panobinostat can reactivate silenced tumor suppressor genes, leading to cancer cell death. These therapies are being explored in various cancers, including multiple myeloma.
Thank you, Aditi, for sharing your insights into the molecular mechanisms of cancer. Understanding these pathways is very crucial for understanding the developmental aspects of targeted therapy.
Thank you, Ritika. It's been a pleasure discussing these topics.
And to our listeners, thank you for joining in on Cancers 101. If you loved the episode, share it, drop us a comment, and let's keep the curiosity alive. Until next time, stay skeptical, stay inspired, and keep exploring.